started. So glad everyone was able to log on easily. Did anyone have any like serious issues with this, or was it pretty straightforward?
Well, pretty much with the code, I would say.
All right, cool. Everyone else doing okay with it? Yeah. Okay.
Yeah.
Sweet. Okay, so I want to start by kind of going through Zoom. Our lecture today is going to be relatively short, so we have plenty of time to kind of go through everything. At the bottom of your screen, there is a chat button. So you can always hit that chat button if you have any questions or if you have a comment or anything that you want to add in. You can just hit that chat, type in your question, whatever it may be, and then I'll be able to see it in real time. So I can just answer it right off the bat right there. So it looks like if you guys have already used it already, so that's good. There's also an unmute button or an unmute button on the bottom left-hand corner. So if you guys, again, if you have any questions about anything and you want to just directly ask, you can always hit that whether you have your webcam on or off. You can just unmute yourself, ask a question or whatever it may be, and then you'll be good to go from there. If you don't want to end up on the video, there's a button right next to the mute button that says stop video or start video. So you can hit that to either activate your webcam or deactivate it. So that's that. I've had quite a bit of time to practice with this whole thing, so it's been it's been crazy. Let's see, so that's that. I believe there's also a button at the top of your screen, or maybe at the bottom as well, that also, I don't know exactly what that button is, but it might be under reactions or something else, that says to raise hand. So again, if you have a question, and you can just raise your hand right there, and then you'll be able to, then I'll be able to, again, identify whether, or who's asking the question, or who's confused, or whatever it may be. And then I can kind of deal with that right off the bat there too. What else? There was something else too, and I forgot what it was. Oh, there is one other thing. So what Zoom does is it will record the class. So right now you might see a kind of a flashing red button at the top left-hand corner of your screen that says recording. So basically how this works is Zoom will record the entirety of our meeting or the entire of our class. And then at the end of it, I will then kind of, it'll download the whole class meeting video thing to my computer. And then from there, I can then cut it down. I can cut out certain parts. I can take different things out, and then I'll upload it onto YouTube. So even if you're not able to actually come to the class live, you'll be able to see the lecture after the fact. Yeah, so I'll upload it to YouTube. I'll take the YouTube link and then add it onto Canvas next to that particular lecture. And I'll kind of show you what that looks like after today. But that should be it. So, so I'll be able to record the lecture. If you're not able to live stream the lecture through your own computer for whatever reason, that's totally fine. You'll be able to watch it later. Or if you do watch it in class, you'll always have the option of rewatching it because it'll be up on Canvas. So there's multiple kind of ways to kind of work through this. I believe that's the majority of the things from Zoom. Any questions on Zoom so far? Kind of going forward here.
No, about the lab maybe.
Yeah, so I'll get to that. In a bit.
Okay.
Cool. Any other questions anywhere? No. Okay. So just want to talk about a few things that are coming up. So today we're going to be going over chapter 5.3. So that's the game plan for today. Again, it's a relatively short lecture. It's not a whole lot to it. And then on this upcoming Tuesday, we're going to have exam 2. Okay, so exam 2 is going to be on Canvas. It's going to be in the quizzes section, just like where your homeworks are at. So the way the exam 2 is going to work is it's going to be active at 8 a.m. And then it's going to close at 11:59 p.m. So you can take the test anytime during that window from 8 a.m. to 11:59 p.m. If you don't take the test, or if you don't submit your exam by 11:59, it will give you a 0. And so you'll end up with a 0 on exam 2. So make sure that you do take the exam during that time period. Kimberly's question, so we're watching the video later, fulfill the attendance. For the attendance, I go based off of the classwork. So once you submit the classwork, that'll count as your attendance. And I'll talk about that in a bit too. Okay. So exam, same format, is still going to have 40 multiple choice questions, 2 extra credit questions, or 2 to 3 extra credit questions. There is no time limit on the exam, so you can take as much time with it as you want, but you only have 1 attempt. Asmin, there should be a button to help you activate the hearing or activate the audio. I'm not sure where that is on my end or on your end of it. Okay, so that'll be the exam. So any questions on exam 2? So again, that'll be up on Canvas. That will be from 8 a.m. to 11:59 p.m. If you don't take it by 11:59, it's going to give you a 0, and then you're going to end up with a 0 on the exam. Okay. So make sure you take the exam. So this will be on Tuesday, this upcoming Tuesday. Yeah. Okay. So homework will go as usual. So it'll still be up on Canvas. It'll still be uploaded on Thursdays, and then we'll still be due on the following Tuesday. So this upcoming weekend, we're going to have homework 8, which covers 5.3. I'll upload that after class today. And then again, that'll be due on Canvas before class. So what I'll do on Tuesday is during our normal class time, we'll have a very short lecture to go over the answers to the homework. It'll maybe be, a 5-10 minute Zoom meeting. We'll go over the homework, and then that'll be it. So you can ask whatever questions you want at that time, or you can, just jump out of it and then just go straight to take the exam, or you can take the exam at some point later on in the day. Okay. But I will go through the homework at our normal class time on Tuesday. And then from there, Thursday, we'll have our normal Zoom meeting with our normal Chapter 6 lecture. So we'll start with Chapter 6 on Thursday. And then everything will kind of go from there. Pretty straightforward. Okay. Classwork. So classwork is going to be up on Canvas. So what I'm going to do is I'm going to hit the share screen button, and you guys will be able to see everything I'm able to see. Okay. So here we go. We will go over to the quizzes section. So we go over to the quizzes section. So now you should be able to see my screen. So this is the part where George takes over. All right, here we go. So once this finishes loading here. Okay, so you will see classwork 319. So that'll be the classwork for that day. And so that is what I'm going to be using to take attendance. So unfortunately, because we're now online, we're not going to be able to do our classwork in groups anymore. So it's going to be individually done. You go ahead and just click the classwork for that particular day. Okay. I'll go ahead and kind of preview it so you guys can see what it looks like. Preview. And then from there, you'll see the question. Okay, so this is today's classwork. It's just one simple free response question, and you're just going to write in the answer to this particular question. Okay, so the classwork is going to be due by 11:59 on the day that it's assigned, okay, or the day of that particular class. So today's Calvin cycle. So this classwork is going to be due by 11:59 tonight. So it just needs to be done by midnight tonight. So that is that. Actually, let me see if I can activate the chat button is not coming. Oh, there it is. Okay. Okay. Any questions so far on what's coming up as far as lecture stuff? Okay. All right. So if there's no questions, I'll show you a few other things that we can do as well. But for lab, so lab is going to be a little bit trickier to do because of this whole online business. So what we're going to be doing is kind of on a week-to-week basis. I'm going to upload some instructions on what we're going to be doing for lab that day. So I'll write those instructions, I'll put them up on Canvas, and then whatever that particular lab is going to be, or whatever that particular lab is going to be that is going to be due by 11:59 on your particular lab day. Okay, so if you have lab on Tuesday, your lab needs to be completed by 11:59 p.m. on Tuesday. If your lab is on Wednesday, it needs to be completed by 11:59 on Wednesday. If you're in the Thursday lab, it needs to be completed by 11:59 on Thursday. I will have the pre-lab lectures happening at your normal lab time. Okay? So for my Tuesday and Thursday labs, we will continue to have our pre-lab lectures from 11:20, or starting at 11:20. We'll kind of explain what the situation is, what our class is going to be about. After the pre-lab lecture, then that's going to be it, and then you're just going to go ahead and do the lab on your own, or do whatever the instructions are for the lab on your own is, and then you will submit that by 11:59. So I'll have more information on how to submit those by the weekend. So just give me a little bit of time to figure that part out. But that's the game plan. If you're on the Wednesday lab, I will. Yeah. So the pre-lab lecture will be on Zoom. Yeah. So thanks for clarifying that. So the pre-lab lecture will be on Zoom at the normal class time. So for the Wednesday labs, I'm going to coordinate with Professor Sharmila, and then we'll kind of figure out exactly how that's going to work out. But I imagine it will probably be the same way I'm doing it. So that's how lab is going to work. Okay. Any questions? Are you basically taking over as our lab teacher? Essentially, we're going to be doing the same things in lab. So Amayali's question is, am I taking over as the lab instructor for the Wednesdays? As of now, not really, but kind of. We're going to be doing the exact same thing all throughout lab, and that will be next week. So there's no lab this week, but lab will pick up next week, starting with lab, I believe it's 8, which is mitosis and meiosis. So yeah, that'll be next week. We'll basically be doing the exact same things. She'll be kind of we'll coordinate with each other and try to figure out exactly how that's going to end up working for the Wednesday labs. And once we have information on that, we'll send that out to you guys. Cool. All right. Any other questions on how lab's going to go?
I'm just confused how lab's not going to work that in general.
Yeah. So like I said, I'll have instructions for each individual lab on a week-by-week basis. So there will be instructions posted for next week's lab, and then that'll be your responsibility to complete and submit by 11:59 on your lab day.
Are the labs going to be so complicated? Like,
not really. They're not going to be they're not going to be as complicated because, again, we're going to be kind of doing it on our own. So they'll usually include some videos, or my own pre-lab lecture, or a combination of the two, and then you'll use that to help you with the lab for that week.
Okay. So it's just like using the information you're going to provide us. We're just like completing the labs. Okay.
Right. All right. Okay. Any other questions? Wait a little bit of time. Okay. All right. So I believe that's it. Again, if you have any questions, feel free to email me or feel free to get in touch with me somehow, some way. But I'll be available as often as I can. Last thing, office hours. So office hours are going to be at at my normal times. So Tuesdays and Thursdays, my office hours will be from basically right after lecture through 11 o'clock. I'll keep the same Zoom meeting, so nothing is really going to change. You won't need to log out or leave the meeting and then come back in. I'll just kind of leave it right after lecture, and we'll be able to ask any questions you have from there. Mondays and Wednesdays, those will be from 11 to 12. So I'll send out Zoom meeting invitations for that. And then everything should be pretty smooth from there. Okay. So last couple things. So with Zoom, like I said before, you're able to either stop your video if you want to not have your webcam available, or start the video. But like I said, I'll be uploading these onto Canvas. So these will be hunky dory. So if you're afraid of ending up becoming a YouTube star because of our lecture, you can always just hit stop video, and then you will be able to see you on the video. Okay. All right, so that is that. Okay, so with that being said, what we're gonna do now is we're going to go over the homework. If there's is there any last questions before we go forward here? Before we start. Okay, so if there's no questions, we'll go ahead and start by going over the homework. So I'm gonna go ahead and hit share screen again. And we will open that up. We'll go over to quizzes. All right. Longer than I'd like. But... okay. All right, so how did we feel with this lab homework? I can't see your guys' faces at this point anymore, so you can just type in the chat how we're feeling or how we felt with the homework. All right, feeling good. Okie dokie. All righty. Okay. All right, so we'll go ahead and just go through things just like we normally would. So we'll start off with our definitions. So looking at the autotroph, how would we describe an autotroph? Is everyone able to see my screen appropriately or adequately? Okay. Okay, so starting with an autotroph, how would we describe an autotroph? You can just type it in the chat and we'll go from there. So capable of producing its own food, right. So it's gonna be an organism that is capable of producing its own food. Perfect. So these guys are usually gonna be using photosynthesis to help produce their own glucose, and then they can use that glucose to help produce their energy. Perfect. How about a heterotroph? Let me kind of move this out the way a little bit so you guys can see. So what is a heterotroph? Consumes other organisms, right. So heterotrophs have to consume other organisms because they are not able to produce their own food. Perfect. Awesome. All right, so how about the light-dependent reactions? Okay, what are those? Light-dependent reactions.
It's going to be a group of proteins used in the light-dependent reactions to absorb light energy and convert to chemical energy.
Not the group of proteins involved here. So it's the first stage of photosynthesis, right? So the first stage of photosynthesis is going to be the light-dependent reactions, and the goal of the light-dependent reactions is to form ATP and NADPH. Good, Brianna. So those the light-dependent reactions will have those proteins that will help form the chemical energy, but the light-dependent reactions is the first stage of photosynthesis. Perfect.
Okay.
Okay, what about the Calvin cycle? Right, so it's the reactions of photosynthesis that use energy made by the light-dependent reactions to form glucose. That's going to be what we're talking about today. Cool. So we'll talk more specifically about that. Perfect. How about chlorophyll A? What is chlorophyll A?
Yeah.
Right, so it's going to be the form of chlorophyll that's going to absorb violet blue, red light, and it's going to reflect green. Okay. Perfect. Beautiful. Okay, next one is the thylakoid. So what's the thylakoid? Right, so it's going to be that disc-shaped membranous structure inside the chloroplast. So this is going to be where the light-dependent reactions take place. Okay. So those are those little pancake guys that are stacked inside the chloroplast. So that's going to be where light-dependent reactions will be taking place. Perfect. Okay, what about the stroma? The fluid-filled space, right. So it's going to be the fluid-filled space around each of the granite, which are those stacks of thylakoids. So that's going to be where the Calvin cycle will be taking place. Perfect. And again, we'll look at that in a little more detail going forward too. Okay, the chloroplast. What do we think the chloroplast is?
The organelle where photosynthesis takes place.
Right. So that's going to be the organelle where photosynthesis will take place. Where's that? Where did I see? Oh, here it is. Yeah. So the organelle where photosynthesis takes place. Okay. And the next one we have is the photosystem. Okay. So what's a photosystem? Right, so that's going to be where we have our group of proteins. So the photosystem is that set of the set of proteins that's going to be involved with absorbing light. Okay? And as it takes in that light energy, it's eventually going to be able to form that chemical energy, be able to form that ATP and NADPH. Beautiful. And the last one is that photon. So what is a photon? A packet of light, right? So it's going to be that packet of light energy. Perfect. And so that is going to be what actually stimulates the photosystems during the light-dependent reactions. Perfect. Okay, any questions on any of these definitions here? Okay, all right, cool. So starting with number 2, so which of the following is true about how the light-dependent reactions work? So we get A, a photon of light strikes photosystem 2 and photosystem 1 to give electron the energy to pass through the electron transport chain. We have hydrogen ion is pumped from the thylakoid space into the stroma by protein complexes, building an electrochemical gradient in the stroma. Yeah, C. Okay, so people are saying A, C, a couple. Okay, then I see C, a few A's. Yeah, so majority of people are getting this one. So it's going to be A. So a photon of light that's going to be striking photosystem 2 and photosystem 1 to help give energy to the electron transport chain, or to the electrons running through the electron transport chain. What's wrong with B? There's a subtle thing that's wrong with B. Does anyone have an idea? So the thing that's wrong with B is this last part, which is the stroma. Okay, so that electrochemical gradient doesn't develop inside the stroma. The electrochemical gradient develops inside the thylakoid space. But those hydrogen ions are going to be moving from the stroma into the thylakoid space where that gradient is going to develop. Cool, so that's what's wrong with B. What's wrong with C? Light-dependent reactions form the energy carriers ADP and NADP+. What's wrong with C?
It's NADPH.
NADPH and what else?
ATP.
ATP, right. So light-dependent reactions are going to form ATP and NADPH, not ADP and NADP+. Perfect. All right, cool. All right, good. So that's good. Number 3. So which of the following gives correct examples of autotrophs and heterotrophs? Okay, so we got A, an autotroph would be a rose, and then heterotroph a coyote. B says an autotroph would be an ant, and a heterotroph would be a daisy. C, an autotroph would be some photosynthetic cyanobacteria, and a heterotroph would be grass. Or D, an autotroph would be a rabbit, and a heterotroph would be a bull. Which one is the correct one? Yes, people are getting this one right too. So we got A. So an autotroph would be a rose, and then the heterotroph being the coyote. So autotroph is going to be able to produce its own food. That would be the rose producing photosynthesis. The heterotroph, the coyote, is not going to be able to produce their own food. So it's going to be able to eat something else. Right, so it has been correct. So because plants make their own food. Exactly. Good. All right, Question 4. So which of the following is the correct set of reactants needed for photosynthesis to occur? So A, we got carbon dioxide, water, and sunlight. Okay, B, we got sugar and oxygen. C, carbon dioxide, sugar, and oxygen. Or D, sunlight, sugar, and carbon dioxide. Yeah, so I'm seeing A's. That's perfect. Exactly. So we're going to be using carbon dioxide. We're going to be using water and sunlight, and that's going to help us form glucose and oxygen, okay, through photosynthesis. Perfect. All right, next question. So question 5. So this goes back to chapter 4.5, I believe it is, or 4.4, 4.5, I think it is. So cholesterol synthesized by our cells, the cholesterol synthesized by our cells uses which part of glucose metabolism as a starting point? Is it acetyl-CoA? Is it glucose? Is it pyruvate? Or is it carbon dioxide? Yes. So perfect. So I'm seeing good answers. Yeah, so acetyl-CoA. So remember, so we're taking acetyl-CoA and we're going to take that out of the citric acid cycle, and that is going to be used to help form cholesterol. And so as a result, you're not going to be able to form any ATP at that point if you're removing acetyl-CoA to help you form cholesterol. Perfect. Okay. Any questions so far? No?
No.
All right, cool. Okay. So next question. So question 6. Which of the following is true about pigments? So we got A, pigments will absorb some wavelengths and reflect wavelengths that cannot be absorbed. B, pigments are not really needed by plants. C, every plant has the necessary pigments to absorb every wavelength of light. Or D, the wavelengths of light are absorbed, will also be reflected by a pigment. Perfect. Yeah, so everyone's saying A, so this is beautiful. So this is the reason why, leaves, for example, will appear green. So leaves are going to absorb all the wavelengths of light except for green. Okay? Because it can't absorb green, it's going to reflect it, and as a result, the leaf is going to appear green. Okay? And that's going to be the case for pretty much every pigment. And there's going to be some colors that it's going to be able to absorb, and then some wavelengths that cannot be absorbed. And whatever can't be absorbed will be reflected. Perfect. What's wrong with B? Why are pigments? Why is this part wrong? Pigments are not really needed by plants. Why is B incorrect?
Because if they don't have pigment, they won't have any color. They will not absorb any heat. Right?
That's So I'll tweak one of the words you said. So he's not going to be absorbing heat, but they're not going to be able to absorb that light energy, right? That solar energy that comes from the sun. So if they're not able to absorb that sunlight, then they're not going to be able to perform photosynthesis. Exactly. Perfect, Brianna. Okay. Cool. All right, cool. So we'll go ahead and move forward here. So question 7. So Mario and Luigi went to dinner and ate lots of carbohydrates, including pasta and bread, okay, containing lots of disaccharides and polysaccharides, before they went to save Princess Peach for the millionth time. So which process of glucose metabolism do carbohydrates enter in order for Mario and Luigi to create energy from them? Right, glycolysis. Perfect. Okay, so beautiful. Let's see. So Johanna had a question. So Johanna said the other day her brother left a Coca-Cola bottle outside, and the next day the labeling was yellow. So did the sun absorb the black and red pigments? So it's not that the sun absorbed the black and red pigments, it's more that the pigments inside the labeling on the can were discolored by the sunlight. So because the sun released a lot of electromagnetic radiation, it caused the colors of the can to discolor essentially and become a little bit different. Okay. Oh, the liquid was yellow too. Oh, interesting. That's a good question. I'm not sure why that's the case. It probably added some extra materials into the soda that made it yellow, but that's interesting. I'm not sure. Yeah, really interesting question. Yeah, sounds gross. Don't drink that. Don't drink that. Okay, cool. So question 8. So what happens to the energy carriers that are formed from the light-dependent reactions? So we got A, they enter the Calvin cycle. B, they enter oxidative phosphorylation. C, they prevent oxidative phosphorylation. Or D, they re-enter the thylakoid space. Right, they're going to enter the Calvin cycle.
No me cree?
Perfect. So we're going to take a look at how that actually takes place going forward today. Okay, number 9. So what is the role of stomata? So stomata allow carbon dioxide to enter the plant from the atmosphere and allow oxygen to exit the plant. B, the stomata are the location of the Calvin cycle. C, the stomata produce carbon dioxide for the light-dependent reactions. Or D, stomata help plants produce ATP and NADPH. What do we think? Right. So the stomata, they're going to allow carbon dioxide in the plant, carbon dioxide and oxygen to be exchanged between the atmosphere and the plant. So stomata will allow carbon dioxide to enter if they're open, and then if they're also open, oxygen is going to also be able to exit out into the atmosphere as well. What's wrong with B? Stomata are the location of the Calvin cycle. Why is this one incorrect?
Isn't it the stroma?
It's the stroma. Perfect. Right. So it's the stroma. So again, similar words, but don't mean the same thing. Okay, so make sure we keep that straight for the exam as well. Okay. Number 10. So Streptococcus pneumoniae is the bacteria involved in producing pneumonia. So S. pneumoniae is a facultative anaerobe. Which of the following is true regarding its interactions with oxygen? So we got A, if S. pneumoniae is deprived of oxygen, it would switch from using aerobic respiration to anaerobic respiration. B, if S. pneumoniae is exposed to oxygen, it would die almost immediately. C, S. pneumoniae needs oxygen to live. Or D, none of the above. What do we think?
E.
So I'm hearing B's, I'm seeing a couple A's as well. So the answer to this one is A. So a facultative anaerobe is able to switch from aerobic respiration to anaerobic respiration, depending on how much oxygen is available. Okay, so if there's lots of oxygen present, it's going to be able to use aerobic respiration, go through glycolysis, the citric acid cycle, and oxidative phosphorylation to produce large amounts of ATP. Okay. But because it's a facultative anaerobe, if there's no oxygen available, it's going to be like, okay, no problem, I'm cool. We'll just switch to using anaerobic respiration, and it'll just do glycolysis and fermentation over and over and over to produce smaller amounts of ATP. Okay. So it's going to be able to live in either environment. So that is a facultative anaerobe, is able to switch back and forth between aerobic and anaerobic respiration, depending on the amount of oxygen available. Now, if it was B, okay, well, if something was exposed to oxygen and it would die almost immediately, how would we describe that particular type of bacteria? What would we call it?
Aerobic. No.
Anyone have an idea? So if it cannot be exposed to oxygen, because oxygen is toxic to it, what would we call that guy?
It would be anaerobic.
Close. So it would be considered an obligate anaerobe. So it's an obligate anaerobe, meaning that if it's basically obligated to use anaerobic respiration because it cannot be exposed to oxygen. So oxygen is toxic to those, so we call this obligate anaerobes. Okay.
So would this one now be? Sorry.
Yeah, go ahead.
Would this one be facultative anaerobes because you can switch between aerobic and anaerobic?
It would be facultative anaerobe because it's able to switch between aerobic and anaerobic. But an obligate anaerobe is only able to use anaerobic respiration. So it's basically obligated to only use anaerobic respiration. So it's kind of an easy way to think about it there.
Mm-hmm.
Okay, this one makes sense to everybody. questions. Okay. Okay. All right. Then the last question here. Okay. So all right. So I see a sort of. So I'll kind of explain this one more time. Okay. So there's a few situations here. So there's really three types of bacteria that we can look at. One of them we didn't go over, which is called an obligate or aerobe, which means it has to have oxygen to produce ATP, which we're not going to worry about. We're not going to talk about it. It's not really that important. Okay. The second type is an obligate anaerobe. Okay, so obligate anaerobes cannot be exposed to oxygen. Okay, if they're exposed to oxygen, it's going to be toxic to them. They're going to die. So the only way that they can produce any ATP or any energy is going to be by anaerobic respiration, where it's going to be able to just do glycolysis, because glycolysis itself doesn't require oxygen. And so as a result, it's going to be able to just produce small amounts of ATP over and over and over, just using anaerobic respiration. Okay. So that's the case for obligate anaerobes. Okay. For facultative anaerobes, they're able to switch back and forth. So facultative anaerobes can switch between using aerobic respiration and anaerobic respiration. Okay. So if there's lots of oxygen available, they're going to use aerobic respiration, go through the normal cellular respiration process using glycolysis, oxidative phosphorylation, and the citric acid cycle, okay, all three of those processes to produce large amounts of ATP. And so that's going to be the case if there's lots of oxygen present for facultative anaerobes. If you are a facultative anaerobe and you're not exposed to lots of oxygen, let's say there's not a lot of oxygen in the atmosphere at that time, they say, okay, no problem, we'll just switch to using anaerobic respiration, and they'll just use glycolysis over and over and over until it gets exposed to oxygen, and then it'll switch back to using aerobic respiration. Yeah, so beyond that's called fermentation. Right. Perfect. Okay. So does that kind of clear things up a bit? Okay. All right, so last question here. So what is the wavelength of the following wave? Okay. All right, cool. Good. I'm glad you got it down. Okay, so what's the wavelength here? So here we have some waves. Is this going to be a 2 meter wave? Is this a 1 meter? Is this 5 meters? Or are these 4 meter waves? Seeing 2. Yeah, perfect. So these are 2 meter waves. So how do we know these are 2 meter waves?
I learned this kind of in math, where we start from where it touches, like on the x-axis. So you're at 0.5, and then to do a full wavelength, you have to go under the x-axis and then over the x-axis and then meet again at 2.5.
Exactly. So that's one way you can go about it, right? So you can start, for example, from 0.5 to wherever we see that next, the next similar point. Okay, that's one way that you can do it.
Yeah.
The easier way to kind of go about it is to look at the distance between either 2 peaks or 2 valleys. So like Mario said, so you can either go from peak to peak. So if you have one peak here at 2 meters, the next peak is at 4 meters. That's how you know we have a 2 meter wave. Okay, that difference between the two. Okay. Or you can go from valley to valley. Right. Beautiful. All right, cool. Glad you got it on my Yali. All right, so that is everything for this homework here. Any questions on the homework or anything from last week?
Okay.
All right. Sounds good. Sounds good. Okay. So with that, we are going to go ahead and finish up our picture here on photosynthesis. And so here we're going to be talking about the Calvin cycle. All right. So let's go. Okay. All right. So starting with the Calvin cycle. So again, if you have any questions, feel free to either chime in or type in the chat, and we'll be able to clear up some of those questions. Okay, so Calvin cycle, this is chapter 5.3. So before we get into the Calvin cycle, what we're going to do is we're going to kind of do a quick little review. So first question. So where do light-dependent reactions happen in the cell? Did it happen in the stroma, the mitochondria, the thylakoid membrane, or in the cytoplasm? So Christian says C, Mario says C. Yeah, so these would be taking place, right, perfect. So this would be taking place inside the thylakoid membrane, right? So light-dependent reactions are happening in the thylakoid membrane. So if you remember from last week, the thylakoid are these discs, are these membranous discs. So each one of these membranous discs is one thylakoid, and they're going to be arranged in stacks called the grana. Okay. So this thylakoid membrane is going to be where the light-dependent reactions will be taking place. Okay, perfect. Okay.
Mr. George,
Yeah.
are we supposed to be looking at something? Because we're still looking at the. Well, I mean, in particular, I'm still looking at the homework.
Oh, are you guys all kind of still seeing the homework, or are you guys able to see the PowerPoints?
Yeah.
Oh, okay. Let's see.
I think there's
Okay.
something on the top right corner where you can switch between the video
Oh,
and what you're.
yeah. I think that's what the issue is. So let me kind of go back here. Oops. That's not what I wanted. New share. Okay. All right. Are you guys able to see the PowerPoint now?
Yeah.
Yes.
Yeah. Okay. All righty. Good. Thanks for bringing that up. I didn't even think to switch back. All right, cool. All righty. So I'll kind of go back so the rest of you guys can see this question here. So the light-dependent reactions would be taking place inside the thylakoid membrane. Cool.
See?
Cool. Yeah, so C right there. So here, looking at the chloroplast, okay, we're going to be able to see our thylakoid here. So these thylakoids are these individual discs, and all these discs are going to be arranged in stacks called the granum. Okay. Then surrounding the granum, you're going to have these stroma. Okay. So when we're looking at our light-dependent reactions, we're going to see the stroma over here, and we're going to see our thylakoid space there. Okay. And so this would be the membrane of the thylakoid brain. Okay. All right, so next question. So which of the following are reactants of the light-dependent reactions? Okay, so what are we starting with? And there's multiple answers for this. Okay, so go ahead and chime in and see what answers you're able to come up with. So we got carbon dioxide, we got water, we got sunlight, oxygen and hydrogen ions. We have sugar, ATP, ADP, NADP plus, and NADPH. So what are we?
B, C, G, and H.
B, C, G, and H. Right. So we're going to be starting with water, we're going to be starting with sunlight, and we're going to be starting with ADP and NADPH, specifically in the light-dependent reactions. Cool. So those are going to be what we're starting with to help us begin this process. Okay. Now, what are the products of the light-dependent reactions? Okay, what are we forming here? Again, there's multiple answers for this one too. So I see some common answers here. Okay. So let's see if we can get one more in here. Okay, so a few people are getting this one. So the ones that we're forming from the light-dependent reactions are going to be oxygen and hydrogen ions. We're going to be forming ATP, and we're forming NADPH. Okay, specifically again from the light-dependent reactions. Okay. So we're going to kind of walk through the light-dependent reactions one more time so that make sure we're all on the same page here. Okay. So the first thing that's going to happen is we're going to have a photon of light hit photosystem 2, okay, right over here. Okay. So once this photon of light hits photosystem 2, that's going to excite an electron and free it up so now it's able to move throughout the photosystem. Okay. When that happens, that'll break water into oxygen and hydrogen ions. Okay. So that's going to be two of our products right there. And that's also going to release a few other electrons to kind of help stabilize this whole process here. Okay. But we're going to free up an electron. From there, that electron is then going to move through the thylakoid membrane's own version of the electron transport chain. And it's going to work pretty much the same way we saw with oxidative phosphorylation. Okay, so this electron is going to move through this first complex, it's going to move to another complex, and as it moves through each of these complexes, what's going to be happening is we're going to be moving hydrogen ions from the stroma across the thylakoid membrane and into the thylakoid space. Okay? And so this is going to be happening against the hydrogen ions concentration gradient. Okay? Again, just like we saw with oxidative phosphorylation. Okay. All right, so then the electron continues to move through, and then eventually it hits photosystem 1. Okay. At this point, the electron is moving a little bit slow, so it needs to get kind of re-energized. Okay, so what's going to happen is another photon of light is going to hit photosystem 1. Okay. That's going to re-energize this electron so that it has enough energy to move through these last two complexes here. Okay. Once it hits this last complex called NADP plus reductase, again, don't worry about the name, just worry about what it does. Okay. So what it's going to do is it's going to take NADP plus and a hydrogen ion, and it's going to convert that into NADPH, okay, and combine the two to form NADPH. And that is going to be one of our energy carrying molecules. Okay. That's going to be one of our products that we're going to be forming here. Okay. The second thing that we need to produce is ATP. Okay. And so ATP is going to be produced exactly the same way we saw when we were going through oxidative phosphorylation. So what's going to be happening is we've developed a very high concentration gradient of hydrogen ions inside the thylakoid space. So what's going to be happening now is this hydrogen is going to start to move along its concentration gradient through ATP synthase. And as it moves through ATP synthase, it's going to convert ADP and the phosphate into ATP. And so that will form the second of our two energy carrying products from the light dependent reactions. All right, any questions on the light dependent reactions before we get into the Calvin cycle? Okay. No questions, okay. All right, cool. All right, so go ahead, perfect. So moving forward, okay, so now getting into the Calvin cycle. So at this point, all we've done during the light dependent reactions is we have formed ATP and NADPH. Okay, that's all we've done at this point. We haven't formed any glucose or anything that's gonna be important for photosynthesis just yet. Okay. So all we've done is we've formed ATP and NADPH. And so those are going to act as our energy carriers for the Calvin cycle. Okay. So what we need to do at this point is we need to start producing some carbohydrates. We need to start producing glucose. Okay. And so what's going to be important about these carbohydrates is that they're all going to be made up of carbon backbones. And so that's going to be important going forward. So keep that in mind. Okay. So those are going to have carbon backbones. Okay. We're going to be making glucose out of different types of carbon-made molecules or carbon-based molecules. Okay. So that's going to lead us into the Calvin cycle. So during the Calvin cycle, what we're going to be doing is we're going to be taking energy from ATP and NADPH from the light-dependent reactions, okay? And we're going to be using those to help us form glucose and other carbohydrates through photosynthesis. Okay? So this is the second phase of photosynthesis. So that's the Calvin cycle. So the Calvin cycle is also kind of referred to as light independent, or sometimes called the light independent cycle or the dark cycle. The reason they call it that is because this cycle does not need light in order to happen. Okay. So it's light independent because it doesn't require any light to actually start it. However, it usually does happen with light because you need ATP and NADPH from the light dependent reactions. Okay. So this usually does happen with light, but it doesn't depend on it. This is able to happen in darkness as well. Okay, so another question here. So which structure helps carbon dioxide enter the plant cell? So we got the stroma, the stomata, the mitochondria, or the roots. Let me see. Okay, so I'm seeing a combination of A's and B's. So again, this is, remember, where we have similar words, but different meanings. Okay. So the last few people have gotten this one. So this is the stomata. Okay. So the stomata is going to be the structure that allows carbon dioxide to enter the plant. Okay. The stroma is going to be the, again, that region outside of the thylakoids. So again, similar words, different meanings here. So the stomata are going to be those openings inside the leaf And so that's going to be important to help start the Calvin cycle. So in order to start the Calvin cycle, what we need is for carbon dioxide to enter the plant cell. So the way that's going to happen is carbon dioxide is going to enter the plant through the stomata. And as it enters the plant, it's eventually going to get into the chloroplast, and it's going to specifically diffuse to the stroma of the chloroplast. So we're going from the stomata of the leaf into the stroma of the chloroplast. And that's going to be where the Calvin cycle will be taking place, inside the stroma. So the Calvin cycle is going to take place in 3 stages. The first stage is called the carbon fixation stage. The second stage is the reduction stage. And the third stage is the regeneration stage. And we'll talk about all 3 of these stages and what happens during each of these processes here. So in order to start this whole Calvin cycle, there's 3 molecules that we're going to need. The first one is carbon dioxide, which we talked about already. The second one is going to be a 5-carbon molecule called ribulose bisphosphate, or we usually abbreviate that to RUBP or RuBP. So this is a 5-carbon molecule, and at each end, it's going to have a phosphate group attached. And then the third molecule that we're going to need is an enzyme called Rubisco. So those are the 3 things we need to start the Calvin cycle. So now we're going to go through each of these specific stages and what exactly happens in each one. So this first one is the carbon fixation stage. So fixation just means we're going to be converting some kind of molecule into something else. So specifically here, what we're going to be taking is an inorganic molecule called ribulose bisphosphate, or RUBP, and we're going to be converting it into some kind of organic compound. That's all fixation is. It doesn't mean you're fixing anything. You're just converting it from one form into another. So what's going to be happening during the carbon fixation stage is we are going to take, I'm kind of drew a little drawing here. What we're going to do is we're going to take some carbon dioxide molecules. So we're going to have 3 carbon dioxide molecules and 3 ribulose bisphosphate molecules. So we have carbon dioxide 1, 2, and 3 over here. And then we have our ribulose bisphosphate. Again, we have 1 over here, our second one, and then our third one over here. So 1, 2, 3 ribulose bisphosphates, and then 1, 2, 3 carbon dioxides. So here's our RUBPs. So each of the RUBPs is going to be a 5-carbon molecule. So this this kind of picture is a little bit incorrect, a little bit has a little mistake in it, in that these should be 5 carbons. So each of these yellows is 1 individual carbon molecule. So we have 1, 2, 3, 4, 5 carbons in our ribulose bisphosphate. Again, ignore this first one. This one shouldn't be here. Ignore that one. But at the end of it, on each end, we're going to have a phosphate group, and that's going to be these red guys. So 1 phosphate group on top, 1 phosphate group on the other side. So what's going to be happening during this process? Let me kind of change color here. So what's going to be happening during this process is each of these carbon dioxides is going to interact with each of the ribulose bisphosphates. So we'll take this one, and it's going to interact with this RuBP. We're going to take the second one, and we'll have it interact with the second RuBP. We're going to take this third one, and it'll interact with this third RuBP. Okay. So what we've taken is we've taken these 5 carbon RuBP molecules, we've interacted it with the carbon dioxide, and what's going to happen is that carbon dioxide is going to then drop off 1 of those carbons into the middle of this process. So it's going to add a 6th carbon to our RuBP. And so what we're going to end up having is 1, 2, 3, 4, 5, 6 carbons. 5 from the original RuBP, and then 1 from the carbon dioxide that it interacted with. So that's going to form a 6-carbon RuBP molecule. Again, you have 3 of those. Everyone following me so far? Are you guys all able to see the drawings that I'm doing too on the diagram here? Okay.
Yeah.
Okay, cool. So once we add that 6th carbon from carbon dioxide, The next thing that's going to happen is each of those RuBP molecules is going to split in half, okay, right down the middle, okay, right there. Okay. So once it splits in half, now we only have 3 carbon molecules. We have 6 3-carbon molecules, which we call 3-PGA, or 3-phosphoglyceraldehyde. You don't need to worry about the name, just 3-PGA is fine. Okay. So it splits in half, and so now we end up having 1, 2, 3, 4, 5, 6 PGA molecules, 3-PGA molecules. Okay. And so this whole reaction here, this whole carbon fixation step is going to be catalyzed by an enzyme with a really long, complicated name, but we just abbreviate it to Rubisco. Okay. So Rubisco is the enzyme that that basically facilitates this entire process from beginning to end, this whole fixation step. Okay. Is this all making sense so far? Yes. So Dominic's question, so does Rubisco help speed up this process? Yeah. So it does help speed up this process. Without Rubisco here, this process would take forever to actually have happen. Okay. So again, just to kind of recap, starting with 5 carbon Rubisco molecules. So again, 3 of these 5 carbon RuBP molecules. Okay. Each of those RuBPs is going to interact with a carbon dioxide, and that's going to add a 6th carbon to those RuBP molecules. Okay. Once you have that 6th carbon, it's going to split in half, and you're going to end up forming 6 3-PGA molecules. Okay. Questions so far on the fixation step? Okay. All right, so that is the fixation step. So the next step is going, oh, sorry, question here. I'm gonna clear all this. Come here. Okay, and we'll clear all this. Okay, so question here. So in the fixation step, carbon dioxide reacts with RuBP to form how many 3-PGA molecules? 1, 3, 6, 10, or 1, 3, 6, or 10?
6.
6. Yeah, that's perfect. So 6 3-PGA molecules are going to be formed at the end of the fixation step. Perfect. Okay. So that's going to lead us into the reduction step. So I'm going to kind of move this out the way a little bit over here. So in the reduction step, so the reduction is going to be the next process here. So reduction is a scientific word, more of a kind of chemistry word that just says that one molecule is going to be taking on an electron or gaining an electron. So gaining an electron from either an atom or some other molecule is called reduction of that particular molecule. Okay. So in the reduction step, what we're going to be doing is we're going to be converting our 3-PGA molecule into G3P molecules. Okay. And so what's going to be happening is they're going to be, these PGA molecules are going to take on, they're going to gain electrons from ATP and NADPH. Okay. And that's going to help convert it to G3P. Okay. So what's going to happen here is each of these 3-PGA molecules is going to interact with 1 ATP and 1 NADPH. So, those the ATP and NADPH are going to donate electrons to the PGA, the 3-PGA, okay, and that is going to help form a G3P molecule. Okay. So again, each PGA molecule will interact with 1 ATP and then 1 NADPH. Okay. So in total, we need 6 ATP molecules and 6 NADPH molecules to convert all 6 of these PGA molecules into G3Ps. Now, when we take that ATP, okay, and we use it, it's eventually going to kind of break itself into ADP and a phosphate. Okay, NADPH will end up getting converted to NADP plus and a hydrogen ion. Okay. Those molecules, the ADP and the phosphate and the NADP plus and the hydrogen ion are then going to go back to be used for the light-dependent reactions. And I'll show you what that looks like in a second here. Okay. But again, all that's happening during this process is each of the PGA molecules is going to interact with ATP, with one ATP and one NADPH to form G3P. So give it a kind of a different formation. Okay. That's all we're doing, converting it from PGA to G3P. Okay. Now again, in doing so, we're going to form ADP and a phosphate, as well as NADP plus and 6 hydrogen ions. Okay. Those are going to go back to the light-dependent reactions to help start that whole process over again. So this is what it's going to look like here. So let me kind of go back here. Okay. All right. So when we take in this ADP and the phosphate, they're going to be able to be used again for forming new ATP later on. Okay. And then the NADP plus and the 6 hydrogen ions are then going to be able to be used again inside the stroma to help reform NADPH later. So we're basically just recycling ADP and that phosphate, as well as NADP plus and these hydrogen ion, so you can continue to produce NADPH and ATP. Okay. Does that make sense?
Yeah.
Yeah. Okay. Any questions here so far? Okay. So again, that's the reduction step. Okay. So a question here. So in the reduction step, how does 3-PGA get converted to G3P? So you got A, each PGA interacts with one ATP to form G3P. B, each 3-PGA interacts with one ADP and one NADP plus to form G3P. C, each PGA molecule interacts with one ATP and one NADPH to form G3P. Or D, PGA interacts with one NADPH to form G3P. So I'm hearing C, I'm seeing lots of C's. Yes, this is perfect. So each 3-PGA is going to interact with one ATP and one NADPH to convert it into G3P, okay, that glyceraldehyde 3-phosphate that we saw earlier in glycolysis. Okay, that's the reduction step. Okay, that one's relatively straightforward, not a whole lot going on there. Okay, the last step is the regeneration step. So this is the part that's the most important for for this whole process to take place, or for helping us form glucose during photosynthesis. Okay, so at this point, we have formed 6 G3P molecules. Okay. From 6, 1, 2, 3, 4, 5, 6. Okay. So what's going to be happening here is 5 of the 6 G3P molecules are going to be used to regenerate RuBP from the beginning. Okay. So again, RuBP, 5 carbon molecules. Okay, 1, 2, 3, 4, 5. In order to regenerate, you're going to need a total of 15 carbons. Okay, 5 for one, 5 for another one, and 5 for the third RuBP. So we need a total of 15 carbons. So we're going to be taking 5 of those G3P molecules, which again have 3 carbons apiece, and that will be used to help reform RuBP, all 3 of those RuBP molecules. Okay, so you're going to regenerate RuBP, and in order to regenerate RuBP, that is going to use an additional 3 ATP molecules. So ATP is going to get used up to help regenerate RuBP from these 5 G3P molecules. Okay. That last G3P molecule is going to exit the Calvin cycle, and that is going to be used to help produce sugar. Okay. So remember, G3P is a 3-carbon molecule. Glucose, as we see over here, is a 6-carbon molecule. Okay. So for that reason, this one G3P molecule is not going to be able to, on its own, be able to produce glucose. So what's going to need to happen is we're going to need 2 molecules of G3P to help form one glucose molecule. Okay. So the way that's going to happen is we need to go through the Calvin cycle twice to form one glucose molecule. So going through the whole cycle once will help produce 3 of the 6 carbons that you need for glucose. Going through it again will produce another G3P that will then be able to help produce that full glucose molecule. Okay. So each G3P basically contributes to half of glucose, essentially. Okay. Does that part make sense? Any questions on that?
No.
Okay. Any questions?
So just the Calvin cycle needs to goes to a process twice to be able to generate glucose.
Right. So it needs to go through this process twice in order to produce one single glucose molecule. Oh, there's one thing I forgot to show you guys earlier. So I believe somewhere there is a button. You should see like an emotions button or a snap button or something like that. If you go to annotate, oh, I think that's where it is. So if you go to annotate, so if you go to the top, you can click annotate. There's a stamp button. So let's just say if this makes sense to you, go ahead and go to the stamp, click on a heart or a star or whatever, and just put it somewhere on the screen so I know if I'm making sense. If I'm not making sense, go ahead and put an X on the screen and let me know. So I got one additional heart. Is there any questions on the Calvin cycle, on this regeneration step? No? Okay, so I'm seeing a few stars. Okay. All righty, cool. So that's good. Okay. So in order to form a single glucose molecule, okay, that one glucose molecule, you need to go through this whole cycle twice. Okay. So what that means is that to produce one glucose molecule, okay, you're going to need a total of 12 ATPs and 12 NADPHs during the reduction step, okay, and then 6 ATPs necessary in the regeneration step. Okay. Again, to produce one glucose molecule by going through this process twice. Okay. So that's going to be necessary to produce one single glucose molecule. All right, so, oops, I'll go ahead and clear all this. All right, cool. All right, so just to kind of walk through, oh, actually, let's ask this question first. So what happens to the 6 G3P molecules in the regeneration step? So we got A, 5 of the G3P molecules are used to regenerate RubyP. The last one, it contributes to making glucose. B, 4 G3P molecules are used to regenerate RubyP. The other 2 form glucose together. C, 5 of the G3P molecules are used to regenerate Rubisco. The last contributes to making glucose. Or D, 3 G3P molecules are released as carbon dioxide. 1 is used to contribute to making sugar, and the last 2 are used to generate RubyP. Yeah, so everyone's saying A. Perfect. Yeah, so A is correct. So 5 of the G3P molecules are used to regenerate RubyP. Okay. The last one is contributes to making glucose. Okay, so we'll go ahead and walk through this whole process one last time to make sure this whole thing makes sense. So starting with the carbon fixation step, let me kind of just go back to the fixation step. So in fixation, we have these 5 carbon RubyP molecules. 1, 2, 3, 4, 5. Ignore the 6th one because this one shouldn't be. 5 carbon RubyP molecules. Okay. Each RubyP, or sorry, we have a total of 3 RubyP molecules. Each RubyP is going to interact with a carbon dioxide, and that's going to add a 6th carbon to that RubyP. Okay. When that happens, okay, that's going to split each of these RubyPs in half into 6 total 3-PGA molecules. Okay. From there, and that's going to be catalyzed by the enzyme Rubisco. Okay. From there, we're going to go to the reduction step. And so during the reduction step, each of these PGA molecules is going to interact with an ATP and an NADPH. Okay. They're going to donate electrons to it, and that is going to convert 3-PGA into G3P. Okay, 6 G3P molecules. Okay. At the same time that's happening, we're going to be producing some ADP and a phosphate, and then NADP plus and these hydrogen ions, which will go back to the light-dependent reactions to be recycled so that you can, again, form new NADPH and new ATP. Okay. So once we formed our PGA molecule, or sorry, our G3P molecules, 5 of those G3P molecules are going to be used to regenerate RuBP, okay, using 3 ATPs, an additional 3 ATPs. Okay. The last one is going to be used to help form sugar or glucose. Okay. So going through this whole process twice will help you form one full glucose molecule. Okay. And that is how we form glucose during photosynthesis. Okay. All right, any questions on this process here? Okay, so there's no questions. We'll go ahead and move forward here. Okay, so photosynthesis, we've talked, so this is what we talked about as happening in photosynthesis in plants, because they're going to have the chloroplasts. Okay. Now remember, chloroplasts are an organelle. Okay, so if we're looking at prokaryotes like bacteria, prokaryotes are not going to be able to undergo photosynthesis using chloroplasts because chloroplast is an organelle. Prokaryotes do not have any membrane-bound organelle, so they're not going to have any chloroplasts to do this process. Okay? So what they're going to do instead is photosynthesis is going to be taking place inside the plasma membrane of those prokaryotes. Okay, so like bacteria, for example. Okay. So their plasma membranes are going to have these little infoldings in it, and so if you kind of think of their infoldings, they're just going to kind of be wiggling around. Each of those little foldings of the plasma membrane is going to have some chlorophyll in it. Okay. So again, it's not going to have any chloroplast, which is that organelle, but it's going to have the pigment chlorophyll, and that is going to be used to be able to capture sunlight so that the prokaryotes are able to undergo photosynthesis. Okay. So it's going to be happening a little bit differently. We're not going to worry too much about how that's going to be taking place, but just know it's going to be happening in the plasma membrane, which will have chlorophyll attached to the membrane directly. Okay. All right, perfect. Okay, so that's going to bring us to the energy cycle. So plants go through this whole extensive process of producing carbohydrates as an energy source. Okay. Why would it make sense? So we're going to kind of talk about why we're going to be storing energy as glucose rather than as ATP. Okay. So glucose is a really, really, really good energy storage molecule because it's very stable. Okay. The other energy molecule that we've talked about in the past is ATP. Okay. ATP, as we've talked about, is very, very unstable because of these 3 phosphate groups that are all negatively charged. Okay. Because they're all negatively charged, they're going to all want to repel each other, but they're all held together by these bonds here. Okay. So because they're being held together, but they want to get as far away from each other, that makes ATP an incredibly unstable molecule. So you're not able to store ATP for long periods of time. Okay. Because again, it's unstable, it's going to break apart, and it's going to release lots of energy very, very quickly. But in a lot of cases, what we want to do is we want to be able to store energy for longer periods of time than what ATP can give us. Okay. So the energy is going to be stored within glucose. Okay. So glucose is a very, very, very stable molecule, and all of its energy is going to be stored within these bonds inside of glucose. Okay. And so because glucose is so stable, we're going to be able to store energy for longer periods of time and then be able to use that more effectively and more efficiently later on. So carbohydrates are going to be incredibly important to produce for photosynthesis in order to help store energy for longer periods of time, okay? And then again be able to produce larger amounts of ATP down the road. Okay. Okay. So when we're looking at plants, plants are going to have chloroplasts, which will help them undergo photosynthesis, but they're also going to have some mitochondria, which are going to help them undergo cellular respiration. So what's going to be happening is all of our plants here are going to be undergoing photosynthesis, and they're going to end up forming glucose or other carbohydrates. Okay, so we're going to be taking some carbon dioxide, we're going to be taking some water, we're going to be taking some energy from the sunlight, so solar energy, and they're going to form sugar or glucose, as well as some oxygen. Okay. Okay. Once they form that glucose is then going to get pushed directly into aerobic respiration. So you can then break down that glucose into oxygen, okay, and then form carbon dioxide, water, and then energy in the form of ATP. Okay. So as soon as they undergo photosynthesis to produce that glucose, they're able to take that glucose and funnel it directly into aerobic respiration to help them produce energy. Okay. And so that's going to be where they're able to produce ATP. Okay. And so these are very effective. And so this is why we talk about plants being able to produce their own food and produce their own energy, because all the energy is going to be coming from photosynthesis, okay, in the form of glucose. Okay. And then from there, they're able to funnel it into aerobic respiration and then form large amounts of ATP very, very quickly. Okay. Now, the last thing here is that photosynthesis is also going to be important because it's going to provide animals, okay, with the amount of, or with the oxygen necessary for them to undergo aerobic respiration. Okay. So the way this is going to work is our plants are going to be consuming lots of carbon dioxide, and as they undergo photosynthesis, they're going to start releasing oxygen into the atmosphere. Okay. That oxygen is then going to get picked up by animals and humans, which will be able to use to breathe, and then we'll be able to use that oxygen to undergo aerobic respiration, and then we'll be releasing carbon dioxide. Okay. And that carbon dioxide will then be used by the plant to eventually undergo photosynthesis. Okay. So this is going to be a cycle where animals and plants are going to be really crucial to each other's survival. Okay. Plants are not going to be able to exist without animals, and animals cannot exist without plants. Okay. Because we're going to be producing the necessary things that we need in order to help us produce energy. Does that make sense?
Yeah.
Either give me yes, throw a heart up in there, or a star, or anything like that. Okay. Yeah, okay, cool. Okay, so here's this last slide here. This last slide just kind of is a little schematic that kind of helps show what's happening during this entire process. Okay. So again, carbon dioxide, or photosynthesis, we're going to be starting with carbon dioxide, water, and energy from the sun to help form glucose and oxygen. And so during photosynthesis, we're going to have our light-dependent reactions, which are going to involve sunlight and water. And those are going to be used to help form ATP and NADPH. From there, the ATP and NADPH will then be involved with the light-independent reactions, in other words, the Calvin cycle. Okay. And that Calvin cycle will then be able to form those G3P molecules, okay, which will again help us form that glucose. Okay. From there, that glucose can then be funneled directly into cellular respiration or aerobic respiration. Okay. I'm going to start here, and then from there, it's going to be able to undergo glycolysis, undergo the citric acid cycle or the Krebs cycle, and then from there undergo oxidative phosphorylation using the electron transport chain, and then produce large amounts of ATP. Okay. So in plants, these two processes are interrelated. They're dependent on one another in order to help the whole process take place and eventually help you produce ATP. Okay. Okay. So that is the entirety of our PowerPoint there. Okay. Any questions on anything that we've talked about so far? No? Okay. If there's no questions, that's it for this chapter. So what I'll do is I'll activate the homework. I'm going to leave our the Zoom meeting open for office hours. So if you have any questions, feel free to pop in. I'm going to go ahead and stop sharing this, so I should be able to see all you guys again. Cool. Okay, so Johanna's question. So anaerobic respiration, carbon dioxide breakdowns. So in aerobic respiration. So in aerobic respiration, carbon dioxide, let me kind of reshare this whole thing here. So I'm kind of back here. Okay, so her question was in carbon dioxide, sorry, in aerobic respiration. So carbon dioxide is not really getting broken down. Carbon dioxide is just getting produced into something else. Okay. Or produced from the Krebs cycle, from the citric acid cycle. Okay. That carbon dioxide can then be used to help produce photosynthesis later. So we're not really breaking down carbon dioxide during cellular respiration.
So everything's being used,
we're just
right?
producing it as a byproduct of it, so that we can use it for photosynthesis later.
So everything's just being a cycle. Everything's just being used for its own purpose.
Right. Everything is a cycle here.
Oh wow.
Yeah, it's all a cycle. Okay. Okay, no, cool, no problem. All right, stop sharing. All right. If there's no questions, you guys are good to go. There's a button at the bottom that says end meeting for you to take off if you're good. If you want to stay for office hours or if you have any questions about anything, I'm going to leave this open until 11 o'clock or so, and then I'll be out of here. And then I will see you guys next week for the homework. Cool.
Bye bye.
So where
Thank
can you find the videos on YouTube? So I will post the link for the YouTube video on Canvas. So once I close this out, it's going to automatically download onto my computer, and then I'll upload it onto YouTube, and then from there I'll take that YouTube link and add it onto Canvas. So you should be able to see everything from there.
All right.
Ryan, so yeah, so we will not be having labs until next week. So next week we'll be starting our next lab. This week we're not having any labs. No problem.
Okay.
All right. Oh, and then also don't forget to do the classwork. So the classwork will be due tonight at 11:59 p.m. So make sure that you take care of that. It's only one question. It's a relatively straightforward question. Shouldn't take too long. So make sure you take care of that tonight, and then everything will be good to go.
So the only thing due today is the classwork?
Yeah, the only thing due today is the classwork, and then we will also have the homework that will be due on Tuesday. Yep. And that homework is going to be a very short homework assignment. I think it's only like 5 questions or so.
Okay, sounds good.